Good day, everyone. It is our pleasure to share with you the findings of our work. I'm John P. Paulin, and my advisor is Dr. Francisco C. Franco Jr., and our work is entitled Molecular Insights into the Antiviral Mechanism of Ebselen Inhibitor against the Main Protease, Mpro, of SARS-CoV-2. I can still recall the government declaring a week-long class suspension in the second week of March 2020. And on the following week, the government ordered a community quarantine, which is sort of a lockdown. From there, the different sectors of the society thought of ways for continuity, like work-from-home arrangements and online classes. And in the past few months, with the decreasing number of COVID cases, these restrictions were eased. The culprit is SARS-CoV-2 virus, which is responsible for the coronavirus disease 2019 or COVID-19. The disease was initially reported and was declared a pandemic in March 2020. The SARS-CoV-2 virus belongs to the family of coronaviruses, which are enveloped. Inside is the single-stranded positive sense RNA genome. The SARS-CoV-2 has a receptor binding domain, RBD, on its spike protein. This structure docks to the body receptors to gain entry into cells and lead to infection. Now, let's talk about the genetic material of SARS-CoV-2. Shown here is ORF1AB. This encodes for two overlapping polyproteins, PP1A and PP1AB. This is cleaved into 16 non-structural proteins, NSP. These proteins may play a role in infection of the cell during virus replication or act in the regulation of the virus replication or virus assembly. It is the main protease, Mpro, also referred to as 3-cell Pro, and the papain-like protease, Mpro, which are responsible for cleaving the polyproteins into the different non-structural proteins needed by the virus. You can see in the slide that Mpro, we have here the red arrow, has several cleavage sites. Indeed, it is important in the processing of the NSPs. Now let's take a closer look at Mpro. Shown here is the Mpro homodimer based on the X-ray crystal structure with PDB ID of 6Y2E. We can see that each protomer has 3 domains. The catalytic dyad consists of a histidine residue, His41, and a cysteine residue, Cis145. This is indicated by a star on each protomer. Now, recall the polyproteins. We have here P5 to P5 prime. These are the amino acids of the substrate that is recognized by Mpro. Now, cleavage occurs between P1 and P1 prime. We can see that cleavage happens between QS, QA, or QN of the polyprotein. Glutamine in P1 is highly conserved in all polyprotein cleavage sites. And in P2, you have hydrophobic amino acids with preference for leucine. And in P1 prime, we have small residues like serine or alanine. Now, currently, we have vaccines which are administered to the population to help fight the infection, preventing severe illness, hospitalization, and even death. Now, the popular vaccines are given as 2 separate doses about a month apart. then booster shots are given for added protection. Nonetheless, there is still a demand for a drug to treat the disease. We may want to look for existing drugs and test their activity against SARS-CoV-2. The advantage of repurposing drugs is that their biological activity as well as their safety profile had been established. In our work, we use Ebselen. This is an organoselenide inhibitor. It was reported to exhibit antioxidant, anti-inflammatory, and therapeutic potential in neurological disorders, cancer, and others. This drug was also found to inhibit other cysteine proteases from other viruses. And as for SARS-CoV-2, Ebselen can inhibit Mpro with an IC50 of 0.670 micromolar. It also has an EC50 of 4.67 micromolar in viral E6 cells infected with the virus. Now, with this information, we are interested on how the ligand interacts with the receptor. This is typically hard to observe using experimental methods. Therefore, computational methods like molecular docking, molecular dynamics, DFT, QMMM, or combinations of this were employed to determine the antiviral reaction mechanism. And in these studies, they showed that the first step is the deprotonation of the cysteine 145 by histidine 41. And this is followed by the formation of a bond between the sulfur atom and that of the ligand or the drug. And this is followed by the cleavage of the ligand. And of course, the regeneration of cysteine 145 and histidine 41. Now, this study employs a semi-empirical method, PM6D3H4, in a truncated model of the protein containing only the reactive residues and is carried out in a gas phase. The computations were done at a much lower cost as compared to other quantum mechanical calculations. The semi-empirical quantum chemical calculations were carried out with the AMPAC-10 package. The structures of the reactant and the product were used as starting and final structures for the CHN calculations. This is to determine the transition states between the starting and the final structures. Then we did also force calculations to verify the minimum and maximum points. The Gauss View 6 program was used to generate the starting structures, input, and to view the output. These are the structures of the compounds used in this study. Now, let's consider first the reaction mechanism of Ebselen with that of the active site. Shown here is the energy profile of the reaction mechanism. And then we have here the structural configuration. Now, these are the bonding pairs that we are interested. We have the SH of cysteine, this NH of histidine. We're also interested with the sulfur-selenium bond. This is between cysteine and that of Ebselen. Then we have the selenium nitrogen of Ebselen. And we have another NH. This one is the nitrogen of Ebselen. Now, at node B, we found that the SH bond in cis-145 was broken. And the proton was transferred to his-41. The SH bond has a distance of 2.32 and the NH is 1.07. The energy involved is 18.7 kcal per mole. Next, the sulfur atom leaves the proton and attacks the selenium atom. This is shown in node C with an energy of 19.1 kcal per mole. The SH distance is 2.73 and the S selenium becomes 2.91 from 3.73 in the previous configuration. At node D, with an activation energy of 36.7 kcal per mole, the selenium-nitrogen bond of Ebselen breaks as the selenium-sulfur is formed. The selenium-nitrogen bond is 3.17 from 1.88 in the previous configuration. And we have the selenium-sulfur bond, which is 2.26 from 2.91 in the previous configuration. Then the selenium-nitrogen bond is fully broken at node E. The distance is 3.55. We also found that the nitrogen of histidine to hydrogen starts to increase, and the nitrogen of Ebselen to hydrogen starts to decrease. This is from 3.83 to 2.55 in this configuration. The activation energy is 38.03 kcal per mole. Lastly, the product is formed wherein the nitrogen of histidine is deprotonated by the nitrogen of Ebselen. See that the NH is 2.88 for histidine and the NH of Ebselen is 1.02. The selenium nitrogen of Ebselen is broken, that is 3.65, and the selenium sulfur is 2.28. We also observe if the changes in the molecular structure of Ebselen may affect the reaction mechanism. Now, in the next slides, we'll talk about the reaction mechanism of EB2-1 and Mpro. The difference between Ebselen and this derivative is the presence of amino group at the phenyl ring of Ebselen. Now, similar to Ebselen, the first step is the deprotonation of cysteine 145 by histidine 41. Then next, the sulfur atom attacks the selenium atom. So we have here the sulfur selenium bond becoming 2.91 from 3.49 in the previous configuration. Now, this is followed by the breaking of the selenium nitrogen bond in Ebselen. Initially, we have 1.88, and then now we have here 2.86. Note also that the sulfur selenium bond is closer at 2.26 from 2.91 in the previous configuration. Lastly, we can see the deprotonation of the nitrogen of histidine by the nitrogen of Ebselen. Basically, the mechanism between EB2-1 and Mpro is similar to that of Ebselen. Now, based from these results, we found that the mechanism of Mpro inhibition by Ebselen is through a multi-step process, starting with the deprotonation of cysteine 145. Then the sulfur of cysteine 145 will attack the selenium of Ebselen, and then the selenium nitrogen bond will break. And lastly, the nitrogen of the Ebselen moiety will be protonated. This has been our work on the mechanism of the Ebselen inhibitor against the main protease of SARS-CoV-2. Thank you very much for your attention. A pleasant day to everyone.